In this video, we will introduce our paper about the framework for automated dissection along tissue boundary. We've turned our attention to automating dissection because despite its frequent use in surgical procedures, there is a significant lack of research on its automation. For our study, we specifically looked at cholecystectomy given the pronounced anatomical differences between the liver and the gallbladder. One of the primary challenges in this task is the limited sensor availability, with the Da Vinci stereo endoscope being the sole sensor. This means the robot has to interpret and locate tissues and instruments in both the image plane and 3D workspace using just the endoscoping images. Furthermore, the robot must operate with some millimeter precision to guarantee safety and avoid potential harm to surrounding tissues. Utilizing Segment Anything, we've accelerated the labeling process, achieving 400 annotations daily per person. These samples demonstrate the speed and the precision on both chicken and liver specimens. To tackle the challenges, we adopted Dissection 2, a model initially crafted for segmenting common objects and detecting human key points for post-estimation. With our custom dataset, comprising segmentations from the previous slide and key point annotations for surgical instruments, the robot can accurately track tissue boundaries and instrument positions through endoscopic images. Furthermore, by merging Dissection 2 outputs with point clouds, we achieve precise mapping of points from the image plane to their coordinates in 3D space. Additionally, we introduced our own calibration method for the Da Vinci system using fiducial markers. Combined with Dissection 2's key point detection, this allows us to accurately estimate the instrument's pose. The robot now receives visual feedback based on the distance between the instrument and its current trajectory point, enabling it to move smoothly along the trajectory while continuously updating its distance. In this slide, we demonstrate the ex vivo procedures. The top row highlights experiments on chicken specimens, while the bottom row focuses on pig livers. On the left, you'll see the endoscopic images taken during the procedure. The center column presents the generated point clouds with the corresponding trajectories displayed on the right. For safety, we've set an initial green point as shown on the right figure. The blue points outline the trajectory based on our segmentation results, and the small red dots indicate the instrument tip's movement along these points. We conducted ex vivo evaluations on 5 chickens, each with varying skin shapes, and 6 livers with different orientations. Each procedure was completed in approximately 2 minutes, achieving sub-millimeter precision.